Hello guys. So in this lecture we will discuss about what is protein and how it is secreted inside the cell. So we will start with the DNA. As all of us know that DNA is a double helical structure. But what is going inside? What DNA is made up of? So DNA is made up of four types of nucleotide. That is A, T, C, G, and if in one strand there is A, then it should always bind to its complement strand, that is T, and T will always bind to its complement, that is A. Similar is with the C. C will bind to G and G will bind to C. These A, T, C, G are called nucleotides. So, and these are the fundamentals, basic nucleotides of which DNA is made up of. And the next step in order to secrete a protein is this DNA will convert into RNA. It is DNA will convert into mRNA. To be more specific, it will first convert into mRNA. And there is one change in the nucleotide that will happen in this process. That is C. I can say C will change into U. Suppose one strand of DNA is replicating into RNA, and that is A, T, C, G. This is DNA, and when it will transcribe, this process is called DNA to RNA is called transcription. So when it is transcribed to RNA, everything remains same as in DNA except T. A will convert it into U, T will convert it to A, C will G, and G will C. As we can see that there will be no thymine. ATCG nucleotides are called. A is adenine, T is thymine, C is cytosine, and G is guanine, and U is called uracil. So let's not go deep into this step, as our objective is to know how the proteins are formed. So this is called transcription. From mRNA, now this protein will form with the help of ribosome. Protein will form with the help of ribosome. And what ribosome does is ribosome will read the RNA. Ribosome will read the RNA, and for every 3 bases, there is a specific amino acid assigned to every sequence of this base. And in human beings, so far in human beings only 20 amino acids are found. So every combination of these 3 bases have 1 amino acid. And what ribosome does is whatever the amino acid is assigned to this code, it will bring that amino acid in this place, and this process will continue till this sequence is completely read out. Even A5. So this process will continue until ribosome read a sequence that codes for stop codon, and that stop codon will terminate the elongation of this chain, and this chain is then this chain based on the type of protein it is been assigned to is called sorry about that, based on the final structure, this chain of amino acid will inherently, because this sequence of amino acid will inherently have a three-dimensional structure, and this this chain of amino acid will form a as soon as this translation process is completed, this chain of amino acid will form a three-dimensional structure based on the sequence it has been translated. And this process of mRNA to formation of chain of amino acid is called translation. Okay, so in next video we will see how the actual shape of the protein will form after this chain of amino acid has been translated, or it is or the shape of proteins is already also been controlled at the time of or processed at the time of this translation. So we will see this in the next video. Thank you all of you, and you can provide a comment about how to improve the contents and the communication in a way that is much more understandable to general public. Okay, thank you.